Executive of Cell Science Conference. Thanks for connecting Cell Science Webinar Edition 1. I request all the participants to mute their laptops or mobile phones in order to have a clear audio of the speakers. On a special note, I would like to all our collaborators, especially UNESCO, UniTwin, Network on Research and Postgraduate Education in Biophysics, Biotechnology and Environmental Health, and St. Joseph God Hospital, Italy, and Journal of Current Biomedical Report. Thank you. Me, along with my manager, Mr. Krishna, would be happy to assist you in this webinar. Hello, good morning, everyone.
Good morning, Apis. Good morning, everyone. Myself, Krishna, manager for the Confrontiers Conferences LLP, and I would like to thank you all for connecting here for the Cell Science webinar for the day one. So, myself along with Alice will be supporting you for the whole day. If you have any problem, you can directly contact via chat, or you can directly let us know through via email or anything else. Thank you, Alice. Over to you.
You too.
Hello. Can we start?
Okay, I would like to introduce Dr. Sinerik Ayrapetyan as the chair of the day one, and I like to introduce him. Sinerik Ayrapetyan has received his PhD in cell biophysics in the Institute of Physiology of Ukraine Academy of Sciences, Kiev, during the period of 1966 to 1970. Currently, he is the coordinator of UNESCO Chair at the Life Science International Postgraduate Educational Center, Armenia. Thank you. And I would like to introduce his title, The Metabolic Drinking Water Efflux from the Cells has a Potential Therapeutic Target.
Thank you very much for introducing me. I would like to say good morning for European colleagues and afternoon Asian colleagues. I hope that you and your family is safety, and we will try to find out the real way to promote our research in increasing organism resistance to so crazy virus as a coronavirus. I think that before starting my talk, I must present a common view about protective reaction of organisms. As you know, any biological systems is open thermodynamical systems which is in environmental medium non-equilibrium state. And the level of non-equilibrium state, it's well known, is determining by metabolic activity of the cells. As you know, organism receive a very weak signal and answer this by very quantum mechanical portion of energy. So it's very important to find out which is the primary mechanism leading to decrease cell or organism to affect environmental factors directed to change this equilibrium state of organism. Please, can I have a slide? Can I have slide?
Alice, can you please share this PPT of Dr. Sinerik?
Yes, sure. Just a minute. Give me.
Yeah.
As you know, there is a great effort, international life sciences community to increase to fighting against coronavirus. But unfortunately, until now, we didn't know by which mechanism virus could penetrate through the cell membrane, which is a border between environment, cell bonding medium, or inside of metabolism. Unfortunately, the main barrier for understanding this mechanism is a traditional old knowledge based on Hodgkin-Huxley theory, membrane excitation, according which the close talking between cell metabolism with external cell bonding medium is realizing by ionic channels, activation and inactivation of this, and the graded driving ionic currents is a main criteria, mechanism through which signal transduction is realizing. In my talk, it's presented absolutely new approaches that signal transduction, close talking between internal and external metabolism is realizing not ionic currents, and the ionic current just consequence of changing cell hydration. As you know, cell membrane is highly permeable for water ions or water molecules, and internal osmotic activity is a higher exceed that extracellular one. So please, next slide. Please, next. This is a simple presented picture of and the previous one, Hodgkin-Huxley model, according which just thermodynamical potential is a tool for exchange information. But from this point of view, it's impossible to explain quantum mechanical sensitivity as well as the response of these weak signals. And now, in spite that in clinics, there are a lot of data that by using extremely weak signal, the physical and chemical signals could be used for therapeutic purposes. But which is the target? Such quantum mechanical sensor in the cell. Next slide, please. In this slide, you can see that aesthetically induced current or any transmitters, any physical factors induced current, water clamp experiments, you can see that magnetized physiological solution treating magnetized preliminary treating by magnetic field depress this transmitter induced current, and after washout by normal physiological solution, you can see that this response is returning back. So in slide B, part B, you can see repeating same experiments when temperature external medium was cold, about 12 degrees. And we have no such magnetized solution effect on this response and absolutely no changing. From this data clearly indicate that in cell membrane, there is a quantum mechanical sensitive mechanism through which can modulate membrane sensitivity, cell excitability, functional activity of the cell. Same thing, it's happening if we will use extremely low concentration biological active substances which are unable to activate ionic channels in the membrane, but could modulate a functional activity of the cell. The fact that there is a net water efflux from the cell, which of course it must be driving, metabolic driving water, because intracellular osmotic pressure exceed extracellular one. It was discovered more than 50 years ago. Please, next slide. Please. In 69, when was discovered that electrogenic sodium potassium pump in normal living state generate membrane current. It means that pushing more sodium and accumulating potassium, less potassium, and pump being high ATP utilizing machine, it could stimulate oxidative phosphorylation processes also, which accompanied by generation endogenous water molecules into the cell. This discovery was independently in our laboratory in Kiev and Gorman Marmari years ago, independent our work was discovered in other mollusks. And finally, in 72 was proved the existence of such generating water efflux from the cell in all experimental model. But unfortunately, before recent years, we and other colleagues, we didn't pay adequate attention on physiological meaning of water efflux from the cell. And it is known that during oxidative phosphorylation processes, please next slide. Please next slide. During the oxidative phosphorylation processes, there is a increasing water. Sorry, before this. Sorry, before this. Okay, stop this. On this scheme, you can see Mitchell theory, who has Nobel Prize because of he find out that for ATP synthesis is necessary to generate membrane potential on mitochondria. In this scheme, you can see that inner mitochondrial membrane, there is a oxidation accompanied by decreasing about 36 molecule water inner membrane. What happens with these waters? Of course, water must push out from the cells, and later we have shown on squitaxon, on other object, that water efflux from the net water efflux from the cells is a key mechanism for controlling low permeability osmotic active particles into the cell. And even mitochondria having such auto-regulation systems that electrogenic hydrogen pump pushing hydrogen from the cells, generating electrochemical gradients on the membrane for these to keep this potential and increased electrogenicity. Of course, it's necessary to increase membrane resistance for hydrogen back. And only when potential was increased rather high, then we have already driving hydrogen inside and synthesize ATP. It is the same thing. It can be demonstrated on mitochondria, on microbes, on plants, and in mammal cells, as a human being also, we have another mechanism controlling low permeability membrane for inward driving osmotic diffusion different substances. It is known that electrogenic sodium pump generating by generating sodium gradient on the membrane, as I was mentioned before this, it generate water efflux from the cells, and in case impairment of sodium pump activity, then reversing water efflux into water influx, which has activation effect on sodium currents and other osmotic currents through the membrane. Please, next slide. So our suggestion is different from traditional suggestion who consider Hodgkin-Huxley models, according which primary mechanism, signaling mechanism is ionic fluxes through the membrane. We suggest that ionic fluxes is consequence of water efflux reversing in water influx. And water influx is well known that any ionic channels containing 3 or 4 molecules water, and only moving this water inside could open ionic channels in the membrane. But from this point, please, next slide. From this point, it's very difficult to explain role cell metabolism in controlling low permeability, semi-permeable properties of the membrane. It is well known that this function of metabolic activity leading to losing semi-permeable properties of the ions. Unfortunately, this dogma about ionic channels is a main barrier for understanding the real protective function of the cell, because on this slide, you can see that well-known transmitters acetylcholine concentration in extremely low concentration from picomole to high concentration, and sodium efflux from the cell. This is a normal living state, sodium efflux from the cell. Only transmitters concentration higher than 10⁻⁷ molar could generate membrane current through the membrane. But before this, before channel activation, you can see that there is an increasing sodium efflux from the cell. In very low concentration, first activate sodium efflux from the cell. It is well known that sodium efflux from the cell could realize only by electrogenic mechanism, by sodium-potassium pump or sodium-calcium exchange in reverse mode. So this mechanism generate water efflux from the cell. And after long incubation in these low concentration substances, you can see that 10 minus 11 molar acetylcholine could depress channel activated current through women. So from this data, and there is a abundance data which were already published, more than 200 research articles in different journals. We demonstrate that before activation ionic channels in the membrane, there is a high mechanical sensitive metabolic mechanism, which response of these activate water efflux from the cell. Now let's find out which is the quantum mechanical sensor through which realizing this stimulation water efflux from the cell. On this figure, you can say that sodium efflux from the cell concentration depend O/N. Different concentration O/N. You know that O/N is another dogma that considering O/N specific inhibitor for sodium ATPase, and for study pump activity immediately using O/N effect. But you can see that O/N only higher concentration 10 minus 7 molar could inactivate pump activity, was sodium efflux from the cell. It is, and there is a small concentration by inactivated sodium efflux from the cell, which is absolutely has no any correlation with pump activity. I would like to remind you that Hodgkin-Huxley theory, which was developed first Peter Becker from Cambridge when he was student, he suggests intracellular perfused axon when was intracellular metabolism absolutely removed from this. Because of this, they could not record this low concentration activation part sodium efflux from the cell. When we repeat this experiment with Professor Peter Becker on intact neurons, we find out that really, in spite that perfused axon gives very important progress in understage generation action potential, but they have big mistakes and they losing intracellular metabolism, which has a key role for controlling cell membrane permeability. Right side, you can see O/N binding, dose-dependent O/N binding with cell membrane. It is a sum of our previous study. You can see that there are two saturated and one linear components, and more detailed investigation have shown that the first saturated components is due to cyclic GMP dependent sodium calcium exchange in forward mode, which pushing calcium ions from the cells. And second part is cyclic dependent sodium calcium. After high concentration, or by you can see that there is a dose-dependent linear uptake cobain, which saying that obain penetrating through the membrane. I'm sorry, this is three family course in hypertonic medium, in isotonic, and hypotonic medium. You can see that all these components is changing by cell swelling and shrinkage. Next slide, please. If we can use, sorry, it's in Russian, it's DNA double RNA penetration through the membrane. You can see that before penetration RNA, again, we have activation water efflux from the cells. This is a well-known model to study pump activation, electrogenic sodium pump. And you can see that if we will apply RNA, again, before penetration RNA in the membrane, cell responding activation electrogenic sodium pump. So water efflux from the cells. More detailed investigation in next slide, please, have shown that, next slide, please. In this case, you can say that this number of receptors or ionic channels is a very dynamic parameter in the membrane, and in response for any increased membrane permeability, increased number of cyclic GMP dependent or cyclic IMP dependent sodium calcium exchange. And in this case, this O-binding concentration left sides, in normal Ringer solution, you can see that acetylcholine induced membrane permeability leads to increased number of GMP activated sodium calcium exchange. Is a normal Ringer is not depend which kind substances. For example, GABA, which is inhibiting transmitters, again, you can see that increasing this number compared number of this receptor in, sorry, receptors membrane. It's also pentylene tetraazole, which well known that activate potential dependent ionic channels, is also increasing response of this, increasing this GMP-dependent sodium calcium exchange. By study, please, next. In this case, you can see that of course, it's well known that water efflux in young animals, it must be higher because metabolic activity is higher. This is in old one. Is water content age-dependent brain cortex tissue after interperitoneal injection, PS, ovoid containing 10 minus 9, 10 minus 4. And previously, from this right side, you can see that ovoid 10 minus 9 has stimulating water efflux from the cell, which is, of course, cannot be by activation pump. And this effect in old is age-dependent weakening characters. But when pump inactive state, when depressing protective function of the cell, we have opposite effect, water uptake increasing. And the right side, you can see that the activation 10 minus 9 ovoid, which was shown, stimulate cyclic GMP-dependent sodium calcium exchange in a reverse mode. There is stimulate calcium efflux from the cell. Calcium uptake. And calcium uptake upon the effect 10 minus 9 ovoid, much higher than when pump in active state. And these processes age-dependent weakening characters. So what does mean aging and aging dependent? It is well known that aging leading to all type risk cell pathology. Okay, next slide, please. It's more detailed. It's by intracellular perfuse method can inject different transmitters in this current, and this response can be depressed by cyclic IMP changing and so on. Next slide. Now what happens with cancer or cell pathology, virus infection. It was shown, as you know, NMR tomography, which was discovered by Raymond Damajan from USA, based that cancer cell overhydrated that normal one. If normal cell is concentration water is about 73%, but in cancer cell overhydrated more than 90%. On the basis of this, he suggests diagnostic parameter as overhydration of the cells. But more detailed investigation, our study have shown that the changing cell hydration in healthy and sarcoma carrying mice, you can see that whole tissue overhydrated. This is healthy heart muscle, skeletal muscle, lung, liver, spleen, kidney, and other tissues. Compare in sarcoma carrying mice, we have hydration of this. And this data clearly indicate again that to respond any factors affecting directing to decrease equilibrium state between cell and cell budding medium, cell responding by activating water efflux from the cells. More detailed investigation, please. Please, next slide. More detailed investigations, we find out that the sodium calcium exchange, intracellular messenger dependent sodium calcium exchange in forward mode and reverse mode, in both cases, there can be stimulated by extremely weak physical factors or extremely weak concentration, homeopathic concentration substances, which are unable to activate, to change ionic channels in the membrane. And this, in spite in which direction, forward or reverse mode, in both cases, there is a water efflux from the cell. And so we find out that any cell pathology, dysfunctioning this quantum mechanical sensitive protective cell function. This is number O by binding in cancer cell, and very low concentration chloroquine. It our recent data, usually chloroquine, hydroxychloroquine, using rather high concentration. It's also traditionally coming to traditional medicine that there is a dose-dependent, linear dose-dependent effect substances, and as cell protective function determining by quantum mechanical mechanism, sodium calcium exchange, which depend cyclic GMP dependent and cyclic IMP dependent, is extremely, can be stimulated very low concentration chloroquine by stimulating this new data 10 minus by 10 minus 11 molar cisplatin, which is using for anti-tumor substances. So by using small concentration chloroquine, combination low concentration cisplatin, 10 minus 11. You can imagine this. You can have therapeutic increase, therapeutic effect of this. So please, next slide. From this, in short, presented my talk, I would like emphasize that cell pathology, that primary mechanism cell pathology is dysfunction metabolic driving water from the cells, which brings to changing number of protein molecules in the membrane, which changing membrane chemosensitivity, ionic channel activity, and very important, unfortunately, we have no time to present the membrane lipid fluidity, which is very often out of the researcher, having key role in regulation water membrane permeability for water, which controlling sodium potassium pump. So sodium potassium pump activated by sodium calcium exchange by removing calcium from the cell. By this way, reactivation pump generate water efflux from the cells. If it will be able to protect penetration foreign particles, including virus, then we can have protective state of organism like us, young generation, which was less sensitive for this virus infection that old one, it is non-healthy. Thank you again. Thank you for listening me. And please, I will be happy to answer all your questions and more detailed information about our approaches. You can find out our website, and I would like to invite all of you to use UNESCO UNITWIN network as a common international platform for collaboration, for changing our approaches, which is I think that the main barrier for solving this question existing many dogma in cell pathology. Thank you for attention.
Thank you
Please.
for your talk, doctor. Do you have any questions?
If you have no question, we will continue, and we will listen very interesting lectures. And from this point of our approaches, we will understand next all interesting lecture, which will be presented leading a number of leading scientists in this meeting. I think that we will be, maybe we will have 5 minutes break to have a rest.
Yeah.
And